The first thing we need to know about DNA profiling is its concept. It is called DNA profiling to a process where there is a specific DNA pattern. This DNA pattern is called a profile, and this profile can be obtained from whatever sample of bodily tissue. It can be blood, it can be a nail, it can be hair, it can be whatever sample we can obtain from a human body. This was discovered by Alec Jeffries in the University of Leicester, and he discovered this technique in 1984. Another thing that is essential to understand DNA profiling is VNTR. What does this mean? VNTR is a variable number of tandem repeats, and these are the DNA sequences that can be located at a defined locus. These sequences are like clustered together, and they go in the same direction. Individual sequences are added or removed through replication and recombination errors.
How do we collect samples of DNA? DNA can be prepared from almost all human cells or tissues. Peripheral blood can be easily and relatively painlessly obtained from an arm, a leg, or even an earlobe. Samples can be stored at room temperature for up to a week without loss of quality of the DNA. When collecting DNA samples with a swab, use a clean cotton type swab moistened with clean water if the stain is dry, and concentrate as much sample as possible onto the swab. If a large sample is available, collect 2 swabs from the item.
Gel electrophoresis and southern blotting. Gel electrophoresis and southern blotting are techniques used to characterize DNA samples. Gel electrophoresis allows to separate the nucleic acid molecules of varying lengths in a sample and visualize them. For that, nucleic acid samples are run throughout polymeric gel matrix under the electric field. Because the nucleic acids are negatively charged ions, a natural or base pH in aqueous environment, an electric field can mobilize them. Then southern blotting is when the RNA is transferred to a nylon membrane and is called northern blotting. Southern blots are used to detect the presence of certain DNA sequences in a given genome. DNA profiling is a process where a specific DNA pattern called a profile is obtained from a person or sample of a body tissue. DNA profile is used to identify a possible origin of a body fluid sample associated with a crime or a crime scene. It's revealed in family relationships, and it could identify a disaster of victims. For example, ESR scientists traveled to Thailand to help identify the victims of the 2004 Boxing Day tsunami. A DNA profile can tell the scientist if the DNA is from a woman or from a man, and if the sample being tested belongs to a particular person.